My name is Saravanakumar Duraisamy, and you can remember me as Sara because it's a big name. And and already Dr. Govind C. Prasad has put a lot of pressure on me. So he has asked whether we have to control the disease or not. Okay. And if we control the disease, then the price may low down because it is again based on the demand and supply. When we supply more, then the price will go down. And when there is a demand, when we supply less, then the price will be up. So it's a like operation success, but patient dead. So we don't want to have that kind of situation here. If you see that the diseases, they are mostly occur during the rainy season or during the wet season, and the production is lower, so that the prices are higher. And we cannot speculate that the losses will remain the same in all the years. And so the disease or the occurrence of the disease is known to cause even 80 to 100% losses. If the disease is very serious and if we are not taking any control practices, then there is a possibility for the loss of entire crop. And in that case, where do we get the Christophene? Where do we have the plants for producing the Christophene? So the control practice is always necessary for producing the Christophene. And if you see the significance of this particular disease, then it affects the plant in the field as well as in the post harvest. So we have to understand the disease and we have to understand the problem. And we cannot say that one farmer who is doing very good control management practices will get lot of prices or lot of income, and whereas the other farmers, because of not controlling the disease, they will lose the crop. If you see the difference between the developed and underdeveloped or developing economy, then in case of the developed economy, the difference between the rich and poor will be relatively low, whereas in case of the developing economy or in the underdeveloped economy, the gap will be very high. So we have to see this particular thing with respect to this, the management, with respect to the equality and with respect to the community, and how we are able to stabilize the sustainable production of Christophene, and so that we will be able to produce more, and so that again we have, when we have more, then we have to think about how we devise the strategies to add value to this. How we can think of exporting, as suggested someone or as said by someone, when we have more Christophene, there is a possibility for the export. And now there are several uses of Christophene has been discovered. It has some cosmetics as well as it has some medicinal properties. So there are several things are coming out. So we have to make use of these things so that we will be able to export it. And again, when we export the Christophene, then again we have to think about the quality of the product. That's how the disease again play a role. So that's why I'm here, and I'm going to cover my presentation on understanding diseases of Christophene. Right? Peasant diseases are major constraints in agriculture, irrespective of all crops. If you see the crops, maybe any crop, there is a loss like 35 to 40% loss. And if you are not taking up any control practices, then it can extend up to 100%. And here, so the diagnosis of disease is a very important issue in the management practice. If we properly diagnose the disease and the pest, then we will be able to reduce 50% of the management cost. Most of the time, the application of pesticides or fungicides are decided by the agro shop owners. And most of the time, we do not know whether it is a pest or it is an insect or it is a nutritional disorder. If we go to the agro shop owner and if we get the pesticide and fungicide for rectifying the nutritional problem, will it cure the nutritional disorder? No. So we have to diagnose whether it is a nutritional disorder or it is an insect or it is a disease. And here, with respect to this Christophene, and based on the samples collected by CABI and based on our analysis, we found that diseases playing a major role, and they are considered as one of the major constraints in Christophene production. And simple thing between the nutritional disorder and disease, they are known to occur on single plant or in patch of plants. If you closely observe and if you regularly visit the field, then you will be able to see the diseases that is known to occur on single plant or in the patches of the plant. Then they will known to transfer or spread from one plant to the another plant. Whereas in case of the nutritional disorders will be shown by the entire field. That's a basic thing, you can have it in your mind so that you will be able to observe the things in the field and you will be able to distinguish between the disease and nutritional disorder. Then of course you know about the insects. Insects are always, you can see the insects in the field or in the infestation site. So based on that, you can decide whether it is a disease or it is a nutritional disorder or it is a pest. Then again, the diagnosis, we will be able to identify the disease based on the symptoms. Once the microorganisms, when I say disease is caused by different microorganisms. Like human beings, like animals, the plants are also affected by the microorganisms such as fungi, bacteria and viruses. And here if you see that this Christophene diseases, or the Christophene plants, they are mostly affected by the fungal diseases. So we will see how the symptoms of the particular fungal disease which is damaging the crop and also the biology of the crop. And based on these symptoms, based on the biology, we will be able to think over how we will be able to manage the disease based on its nature, based on its spread, based on its survival. As I told, the fungal diseases are known to affect the leaves, stems and fruits. All stages are affected by this particular disease, and that is called gummy stem blight. In addition to that, there are some diseases like powdery mildew and downy mildew, and also some leaf spots caused by Cercospora and the Colectotrichum. Okay. But here if you see the very important disease that affects the Christophene production on leaves and on stem and on fruit is a gummy stem blight. So from this you can understand how significant this particular disease is, because it is not only affecting the leaves, not only affecting the stems, but also affects the fruits, which is the economically important product. So from this one can understand that the pathogen can survive in the different stages of the plant. In addition to that, we have also observed the nematode infection in the roots. If you see this particular gummy stem blight disease, and when it affects on the leaves, it is named as the bullet hole symptom, and when it affects the fruits, it is named as the black rot. And here the fungus is Didymella brioni, that is the scientific name of the fungus. And here this particular disease is known to occur or known to affect all cucurbitaceous or cucurbit plants like pumpkin, squash and cucurbits and watermelon. So So because of the succulent nature of the plant and leaves, the pathogen is known to affect the plant. And here from the survey, almost we have seen infection in all the leaf samples collected from by Aldo and Dr. Ramnan. So here this is the fungus we have isolated in the lab, and because of its polyphagous nature and the disease can spread or it can present in the atmosphere or in the soil or in the plant debris all the time. So now we will see how to diagnose the disease in the field, because I already told you that the early diagnosis of this particular gummy stem blight disease will solve the or will help to devise the management strategies. Here the bullet hole. Actually I heard this bullet hole name from
the farmers call it.
yeah, okay, that makes sense because because here you can see the hole. Here you can see the hole. When you make a bullet shot, then you can see the spot. So that spot really it makes sense. And here you can see the ash colored small lesions, and initially if you see that, you may assume that it may be the scraping by the some worm or some insects, but it is not because of the insect, it is because of the fungus, right? And later these small ash colored lesions will enlarge and they will join together, then they will produce the blighted conditions, or the leaves will dry, the leaves will dead. And here when you zoom this or when you use some lenses, you will be able to see some black tiny dots inside these ash colored spots, and mostly these ash colored spots, they will start from the leaf margins, and then you can also see the small minute dot colored, which is the seed like structure, which is actually considered as the seeds. Like how the plants or weed plants, they produce lots of seeds, the fungus also produce lots of seeds. And stem blight, and here on stems or on vine you will be able to see the elongated slender ash colored spots. Initially when they brought it to me in my lab, I thought it could be because of some insect scraping on it. Then later when we take the sections, when we isolated the fungus, we understood that this is not because of the insect but because of the fungus. So here probably you might ask, here we are not seeing any gummy or exudates from the stem, but still why it is named as the gummy stem blight. And here if you see, this disease is known to occur on the pumpkin and on watermelon and squash, and when it affects those crops, it is known to affect the stem as well as on the fruits. And when it affects the stem and fruits, it is known to produce some gummy like substances. That is why the disease or the pathogen is named as the gummy stem blight disease, right? And also on fruits you can see this kind of rods. Initially it will be ash colored or slight white color, then later if you keep it in the refrigerator conditions or in the cover with a little sprinkling of the water, then you will be able to see some black colored dots and some mycelium or whitish fungus growing on it. So this is again a major thing in the post harvest. Sometimes you will not be able to see the symptoms. The pathogen may present inside or it may present on the surface of the fruit, which may not express the symptoms, because you know that the symptoms are expressed only when it colonizes the entire fruit or when it colonizes the spot, and when it kills the cells, then the symptoms are expressed. And here you can see the depressed or sunken spots, right? And that's why this is a major problem. When you send it for some marketing or when you export it, initially you may not see this type of diseases or symptom expressions. But when it reaches there after a week time, then they might be able to pick up this disease. Then they may discard it. Then it may tend to a lot of losses. That's why we have to be very careful in managing this disease in the field, so that we can reduce the inoculum, we can reduce the infection in the fruit, and we will be able to produce the quality Christophene. So the occurrence of the disease indicates how significant is on leaves as well as on the vines and on the fruits. So based on the symptoms, we will be able to diagnose this fungal disease. And here, this is how we diagnose in the lab. Actually, the fungus is known to produce the spores by sexual reproduction and asexual reproduction. And that is why if you see the name of this disease or pathogen across the world, there are several names given. Initially, this is actually the asexual reproduction of the fungus, and this is the microscopic field. And here, if you observe this black tiny dots, and this will be like this. And inside there will be millions of spores. Inside this dot structure, you can see millions of spores. And here you can see, here we zoom the individual pycnidia. This is called pycnidia, and here it produces millions of conidia, and each conidia can act as the seed, right? So this is the asexual reproduction. And again, the scientific part of it, I don't want to go in, because then you may feel that, you know, I'm like teaching to the students, so I don't want to take that part. Okay, just is the asexual part of the fungus. And similarly, still if you zoom it, then you can see this kind of structures in the microscope, which is responsible for the infection. And this pycnidia are these small minute dark structures you will be able to see on the dead plants, as well as on the dried leaves, as well as on the infected stems, and as well as on the fruits. In addition to that, the fungus is known to produce another type of spore. This is again another seeds, and this is the sexual spores. And here this is actually called as the didymella, and this spore is produced by the pathogen. So the pathogen, it can survive as the asexual body, and it can survive as the sexual body, right? So now, what are the conditions favorable for the disease development? The first important factor for the development of this disease is the moisture. So when I realized the condition of the cultivation, as well as the humidity and leaf wetness, and here the pathogen is known to infect the fruit or the leaf when the leaf wetness is present at least for 1 hour. And here, when there is a free moisture present on the leaves for 1 hour, then the disease can affect, or it can initiate the infection. Because always the disease occurs during the favorable conditions. Even if you see the human diseases, when the environment condition changes, when the weather condition changes, then a lot of people will get some sick, or they will get some cold, or the same thing applies to the plant. So when there is a rainfall, then there is a humidity, there is a moisture on the leaf, so that the disease is tend to occur, or infection will be there. So that is the major thing. And as well as if you realize that some farmers, or almost 55 to 60% of the farmers from the survey, they are using the sprinkler system. Of course, the sprinkler system, you know that it enhances the leaf wetness. So there is a possibility for more infection when there is a leaf wetness. So so for this we cannot say that, okay, don't use this sprinkler system. We cannot say that. But what we can do is, the sprinklers or the irrigation, it can be done in the morning, so that the dry sun will come up and the leaf may dry up. That is the strategy, or that is the practice suggested. And in addition to that, suppose if the sprinkler or if the irrigation is done in the evening or in the afternoons, then we may not get the sunlight. So in that case, the leaf moisture can present, or the moisture can present on the leaf, and leaf wetness will be there. And when there is a leaf wetness, then overnight the disease can take up the upper hand and it can infect the leaf. So that's why we have to understand the biology, or how it spread, or how it survive. And another thing is the relative humidity, and optimum temperature is almost 25 to 30 degrees Celsius. And here if you see this microclimate existing in the Christophene cultivation, it will be similar to this temperature. But above the temperature and above the relative humidity, leaf moisture or the plant moisture or the leaf, the water droplets or particles playing a major role in initiating the infection. So now we will.
Can I ask a question?
Yes.
I would answer what you just said here, and I would say a sudden fluctuation in temperature. Like if you have hot sun and you get a shower rain, you know that is the disease coming right.
Yeah. Okay. Yeah. Okay. Excellent. Because that's what happened. Because the pathogen, it always present in the plant debris or in the atmosphere or in the soil, but the infection takes place only during the favorable condition. You are right, it will be there, but infection will not be there. But when there is a leaf wetness, because pathogen or the fungus, it also requires some conditions for its growth and development. It always remain as the dormant. Even when we put the seeds, we have to irrigate it, and if we are not irrigating the seeds, then the seeds will not come up. And similarly the fungus, it requires the conditions. Even the disease will not occur all the human beings, and it's again based on the immunity and based on the conditions. This all things applies to the plant diseases. That's why we probably say the persons who are studying the plant pathology can probably call them as the plant doctors. Okay. And here, how does it survive or spread? We have to know that. And here, this can present on the dead plant materials or on leaves, and as well as it can infect the fruits, so that you will be using the fruits for raising the seedlings. And there is a possibility, because when the fruits are infected by this disease, then the inoculum will be there, then inoculum will tend to transfer to the seedlings.
So all we can do when you infected seeds, all we can do is we either use them up, or all we can do is kill them.
That's why you have to select the disease-free fruits. And also we have certain practices at the end of this presentation, and wherein we will be able to see how we integrate things, so that we will minimize the disease and we will be able to minimize the losses, right? And here the pathogen survives between seasons on diseased vines and crop debris and on the weeds. And the next important thing is the pathogen can survive for up to 2 years. I told you that Pycnidia and the ascospores, that is the sexospores, they can survive up to 2 years. So in some of the countries, in some of the regions, the practice is at least they should do the crop rotation. When they do this crop rotation, when they are rotating this Christophene cultivation with other crops for about at least 2 to 3 years, then they will be able to reduce this disease infection. That is a finding. And someone has asked the question whether we will be able to grow this Christophene in the plains. And as if you see the family of this plant, cucurbits or cucurbitaceae, is a pumpkin and is a squash, there is a possibility. And that's how it is practiced in some of the countries, and they are rotating with the crops. So we have to see the possibility. of crop rotation. I already told you that this is spread by the splashing water and by the rainfall. The spores which is present, it will be spread from the infected plant to the healthy plant by means of the splashing water or by means of the rainfall. So when we use the sprinkler, there is a more possibility for the spreading of the disease, as well as you are giving the conducive condition for the infection. So we will see how to manage this disease, this particular disease, and most of the practices, or most of the time, the disease is controlled by administering the chemicals. But in addition to the chemicals, we will see, or we have to see what are the viable or feasible practices we will be able to follow so that we can bring down the disease. The first thing is the crop rotation. If possible, we can rotate this with the other crops, with non-cucurbitaceae crops if possible, because I told you that the pathogen is able to survive in the debris or in the organic matter or in the soil for about 2 to 3 years. So if we keep up, suppose I'm not suggesting this for all the fields, but at least if you have the 100% infection continuously for 1 or 2 years, then this will be the option. If you have the complete loss or the 100% infection, then this will be the solution.
Those are the crops you want to rotate it with, right?
No, this is just for example I have given, and you can. Sorry.
Why not vine crops?
Because same thing,
Probably
same problem.
It will be the same thing.
Which crop?
Vine crops that grow on vines, like
The same problem.
no, not cucurbits. If you put a
okay.
vining legume, for example,
Yeah, you
rotate that?
can rotate with other crops. Yeah, non-cucurbits. You should not grow the karali. Sorry? No, you should not grow the. No, you can karali. You cannot grow.
Karate is the same farm.
Bitter gourd is the same family.
All I same thing, same thing.
No, Barbadine is not the same farm.
Bitter gourd, pumpkin, watermelon, okay, and squash. They all come, cucumber. They all come under the same family, so you cannot rotate.
So like Bodhi and Singh, those kinds of things.
Cucumber,
Cucumber is the same farm, yeah.
Yeah, cucumber is the same family.
I left it without anything. I just left the grass and let it grow up, you know. Get rid of the Christophene and just leave it like grow back a while. And then,
Yeah, okay. When you leave the field fallow, like without any crops, yeah, for a year or two, then what you have to do is you have to incorporate, or you have to plow that grass into the soil before going for the Christophene, so that the debris will not be there. No, you need not to keep the field every time clean. Like if you keep it as a fallow, what I'm suggesting is one time, yeah, before going for the Christophene.
and I feel that it really get bad, and I left it. I get rid of the Christophene, leave it like grow up, and then I cut plants everything back and plant it over and come back good.
Yeah, it can come back. That's what it can come back. But when the conditions are conducive, the pathogen will be there. The same problem will come again. So that's
At least again a good production for a good while without it being so bad.
yeah, that's what. When you redo the cultivation, then you will because the results or some of the studies says that you can get increase in the yield when you do this rotation. Yes, sir.
But say rotate it, right? So when you want to plant back the next 2 years, where you getting the seed then? So
Okay. You have to choose the seedlings.
Huh?
You have to get the seedlings.
Oh, you need to dip them still, although they don't look like they have any disease on it. Like,
Okay.
Still have the pathogen in it.
when I say come into that shop,
Yeah. Okay. You have to get the healthy seedlings.
from somebody else.
Okay, that is again here. Do they have
I think
any?
we should, in terms of time, let's finish the presentation and then we take the questions.
Yeah. Okay. Because in some countries they do the nursery and they sell the seedlings, but I don't know the system here how it goes. But here, you know, from the survey we understood that you collect seedlings or seeds or fruits from the person who has already the crop, and you can
Everybody keep their own seedlings.
Seedlings.
That's the
Yeah.
what happens now.
but in that case, then again you have to be careful when you get the fruits from the field, whether that field is a healthy field or it's a infected field, because that's the problem in case of banana as well as in plantains. When we get the suckers and we are not sure whether they are infected or not.
The infection they're right through. It's just the condition changing.
Yeah.
They're right through.
Yeah. So the next thing is yearly and correct diagnosis. Once the proper monitoring or proper scouting of the diseases, so that you will be able to take up the yearly management practices. Then again, when it comes to the management practices, then this is again, if you see the disease in few plants, then it's better to remove the leaves, infected leaves, and you can burn it, the leaves, so that it will reduce the infection, right? And also I told you about the irrigation, like the sprinkler, how it increases the disease infection. Then the chemical control, and here once you see the disease, you have to follow the crop rotation, or else you have to diagnose the disease, and if it is very early stage, then you can pick out the infected plants and you can burn it, and you can see that it is not spreading, because it always, the initial infection will serve as the source of inoculum for other plants. Once the leaves or spars they are appear, and then you cannot revive the dead cells. Only thing is we can prevent it spreading from infected to the healthy plant.
Wild citron, what's that?
Wild citron, okay, it is actually wild melons. Wild melons. In hills or in forests, you can find the wild type of melons. It is not commercial types. It is not cultivated types. Okay, so if you see those things as weeds, then it's better to remove, because they can also serve as the host for this. Yeah, Karali. Yeah, Karali is a bitter gourd. It's a cucurbitaceae plant.
Yeah, Chingy.
Yeah.
Bush is also,
So the chemicals which could give very good results, or one is the Folicure, that is actually tebuconazole, that is known to effectively control this disease. That is actually marketed in the name of Folicure. Okay. The technical name is different from the commercial name. Okay, because each company like Syngenta or Bayer and everyone, they market in their trade name. But if you see this technical name, this will be common for all the companies. Like the technical name is tebuconazole, but for your understanding, I'm saying this Folicure. It is marketed as the Folicure. Then the second chemical is the chlorothalonil. This is also known to give the very good control of this, and this one is marketed in the name of Bravo. Bravo means, you know, is a good. And mancozeb is again another chemical, that is actually dithane M45, and also there is a combination of mancozeb and copper hydroxide that is marketed in the name of mancozide. So you can rotate these chemicals. I advise you to rotate the chemicals, because these fungicides, they have different mode of action. I am suggesting the chemicals which has the different mode of action, so that the pathogen or fungus, when you administer the same chemical over a long period of time, then it will develop the resistance, then it may become super strain, then whatever chemical you apply, it may not be effective to control that particular thing. Like how we are, if we get the antibiotics all the times, then our body will get resistance. So no chemical will control So we have to be very careful. And another thing is, at least we have to give 7 to 10 days interval to see the results. But sometimes the farmers, or I'm not putting a blame, but sometimes they will go over like within 3 days, like twice in a week, because I already told you that the dead cells, you cannot revive the dead cells. Only thing is you can prevent infection, as well as you can see that new flushes are not getting any infection. So within 3 days or 4 days, again going on the another round will not make any sense, and it will result in the higher investment. Then the possibilities of biopesticides. Sorry, just I'll wind up in 5 minutes. Okay, I think I'm just going over my time. Okay, the biopesticides, and here the biopesticides, actually right now we are having the project that is actually agriculturally important microorganisms project that is funded by UV and TT government, and we are working on the development of some beneficial microorganisms which can promote plant growth as well as control the diseases, right? So right now we have isolated some microorganisms in the different ecosystems of Trinidad, and we have find out that some has very good effect in the lab as well as in the field conditions, just in our trials in hot peppers and in tomatoes, and probably we need to work on this more with respect to this disease, Christophene disease, and we could see how possible we can develop as the biopesticide for the control of these diseases. So in that case, even when we have the biocontrol agents, even before marketing it, we will be able to dip it into the biopesticide solution, and we can send it for the marketing, and we can send it for the export. That's what we assume, and we are going to work on it. Okay.
I think this is a living organism, right? You can develop something to kill it.
Develop something
to destroy it.
Yeah, that's what we are doing it. Yeah. So there are some beneficial microorganisms. We use these beneficial microorganisms to eradicate or to control these harmful microorganisms, like biocontrol agents and biofertilizers. So in addition to the control, this will also help the plant to grow well, and this will increase the plant vigor. I told you that once after harvest, if you have the debris, those things, then it's better to collect or burn it, or else it's better to incorporate in that soil itself, and you have to do the deep ploughing so that it will just go inside. Okay, and also you have to avoid injuries to the fruits when you harvest it. And so by possibly integrating all these practices, we will be able to achieve the integrated pest management and disease management practice to this particular disease. And take home message here, we will be able to use the IPM for the sustainable production of Christophene in Trinidad, and I hope again Dr. Govind is waiting to put pressure on me. So with this, I thank all of you for your patient hearing, and I don't know, some of you might be new to my accent, but I hope everyone would have understand it. Yeah.